Hi, Dr. Javed Akhtar Ansari. Welcome everyone in this video lecture. I am going to talk about gene expression and its regulation. After watching this video, you will be able to understand the central dogma of molecular biology, gene expression, what are the different types of genes and gene regulators, and the gene regulation in prokaryotes and the gene regulation in eukaryotes. We try to understand the concept right from the basic, that is central dogma of molecular biology. So, central dogma of molecular biology consists of replication, transcription, translation. So, what is replication? DNA makes a copy of DNA by the enzyme DNA polymerase known as replication. Then comes transcription. From DNA make RNA by the enzyme RNA polymerase, that is transcription. Then comes translation. From RNA, protein is made by ribosomes known as translation. See here. Coming to the next point, what is gene? So, gene is a part of DNA and DNA is a part of chromosome. Chromosome is a part of nucleus, which is a part of cell. It is necessary that protein must be synthesized in cell at the proper time to function properly. All the cells control or regulate the synthesis of proteins by using the information encoded in their DNA. So, the process by which a gene produces RNA or protein or both is called as gene expression. So, what is functional gene products? What are the functional gene products? So, there may be protein and there may be sometimes non-protein as well. For example, insulin and chain of hemoglobin, etc. These are the protein products. And non-protein includes the functional RNA such as ribosomal RNA, transfer RNA and sRNA. So, what is gene expression? So, now we try to understand with the help of example. For example, insulin gene synthesis is produce or express functional gene protein named insulin when the level of glucose is higher by the process of transcription and translation. So, functional gene product is not always protein. It may be non-protein coding genes, for example, RNA, tRNA, snRNA, etc. So, here when there is increase in the blood glucose level, so at that time insulin is required. So, in this situation, insulin genes get activated and followed by transcription and translation, there will be synthesis of insulin or the expression of insulin. In different cases, in different genes, transcription takes place and messenger RNA of beta chain of hemoglobin is formed, followed by translation, and the final product is protein, that is beta chain of hemoglobin protein here. So, as and when required, the particular gene process is initiated and that particular gene is expressed. Means all the genes are not expressed at all the times. Now comes to the types of genes. Generally, genes are basically of two types. One is constitutive or housekeeping genes, also known as constant genes. So, and another one is regulatory gene or inducible gene. The constitutive genes, this type of genes are always active. Their expression is not regulated. So, these genes expressed at a constant rate, for example, enzyme of citric acid cycle. Another one is regulatory gene, that is inducible genes. It is expressed only in special circumstances. For example, genes for gluconeogenesis will be inhibited in normal situation, that is after food. But these genes are activated during fasting or starvation. Then comes to gene regulators. Regulatory genes can be of two types again. One is positive regulator, also known as activator or inducer. Another one is negative regulator, also known as repressor, silencer or inhibitor. So, what is positive regulator? Here, gene expression is increased by a regulatory element. In negative regulator, if gene expression is decreased by a regulatory element, it is said to be a repressor or inhibitor. Then we try to understand the gene regulation in prokaryotes. As we know, the prokaryote organisms are single-celled organisms that they lack a cell nucleus and their DNA therefore floats freely in the cell cytoplasm. To synthesize a protein, the process of transcription and translation occur almost simultaneously. When the resulting protein is no longer needed, transcription stops. As a result, primary method to control what type of protein and how much of each protein is expressed in a prokaryotic cell is the regulation of DNA transcription. Therefore, in prokaryotic cell, the control of gene expression is mostly at the transcription level. And now we'll understand with the help of this figure. So there are two scenarios. One is absence of lactose and another one is presence of lactose. So like operon concept. So here in absence of lactose, at left hand side of the figure, operon consists of like I, which is constitutive gene creating mRNA by transcription, then undergo translation and codes for the multiple protein, that is repressor tetramer subunits, which bind to the operator. Now this operator will not allow to bind transcription enzyme, this RNA polymerase, to promoter site here. Therefore, all the downstream genes like Z, like Y, like A cannot be transcripted. This way, when lactose is absent, these like operon make sure that the lactose metabolizing enzyme cannot be synthesized. In situation 2, that is presence of lactose, so like I being constitutive genes always synthesize repressor protein tetramer. So lactose, which contains glucose and galactose, is hydrolyzed by galactosidase enzyme and produces allolactose that will bind to the repressor tetramer, and this repressor tetramer will become inactive, which will not be able to bind to the operator. Thus, the operator will allow bind transcription enzyme RNA polymerase to the promoter site. All the downstream genes like Z, like Y, like A will be turned on. Transcription starts create messenger RNA followed by translation and expressing the proteins. So here, when lactose is present, the expression of lactose metabolizing enzyme takes place. Then comes gene regulation in the eukaryotes. So in eukaryotes, eukaryote cells have much larger genome and much greater cell specialization. In eukaryotes, the expression of genes into the protein can be controlled, regulated at various locations. Eukaryote gene expression involves many steps, and almost all of them can be regulated. Here, the DNA is contained inside the cell nucleus and there gets transcribed into the RNA. The newly synthesized RNA is then transported out of the nucleus into the cytoplasm here, where ribosome translates the RNA into the protein. When gene expression proceeds abnormally, serious imbalances and diseases may take place, including cancer. Eukaryotic gene expression is regulated at many stages. So right from the chromatin structure to the protein activator control. So these steps include regulation of chromatin structure, transcription control, RNA processing control, RNA transport and localization control, translation control, messenger RNA degradation control, and protein activation control. Although all the stages of gene expression can be regulated, the main control point for many gene is transcription here also. So with this I conclude my lecture. Hope you have understood the concept, and will meet again with the more interesting video lectures. So keep watching the videos. Thank you.